The graph of an enzyme-catalyzed reaction progress is very similar to ordinary chemical reactions. To study the reaction progress, there are a few methods that we can use. First, we'll be looking at the enzyme catalase, which catalyzes the breaking down of harmful peroxide in our bodies into water and oxygen. So if you've been observant, you will have noticed that I forgot to put state symbols. And now it becomes clear that this reaction results in the production of oxygen gas. You can of course test it by seeing whether it reignites a glowing splint, but more importantly, you can measure the amount produced with a simple setup like inverted measuring cylinder or syringe. Since the amount of oxygen is representative of the amount of products formed, we can plot a graph of products against time. And it will look something like this. Initially, there is no products and a high concentration of substrate. As the enzymes convert more substrates into products, the substrate concentration is reduced, and the chance of a successful collision with an enzyme molecule is also reduced, slowing the enzyme-catalyzed reaction asymptotically until all the substrate has been consumed. For reactants or substrates, since the production of the products means a consumption of the substrate, it would make sense for the graph to look like this. So if you're ever prompted to draw a graph of products or reactants against time, you know how to draw it. Similarly, for other enzymes, such as amylase, which breaks down starch into simple sugars, it doesn't produce gas, but you can use other methods to measure it, such as like quantitative analysis or spectrophotometric analysis, or simply observe for color changes in the mixture.